This is our third video on Gymnosperms and their naked seeds. In this video, we're going to look at the pines, the pine life cycle within the Panopto, this group of conifers. And then within that group, we have the Gnetophytes. We'll look at the Gnetophytes first because we have a lot to cover when we get to the conifers. So this group is weird. Genetics point to it being this highly divergent lineage within the conifers, but they look a lot like Angiosperms. So before, we thought maybe the Gnetophytes were this transitional group between the Gymnosperms and Angiosperms because they have things like double fertilization, which you're about to learn about. They have flower-like structures, and they have vessel elements, which you do know about, and those evolved within the Angiosperms. So very specific examples of this convergent evolution. So morphologically, we place them closer to the Angiosperms, but genetically, they fall within the conifers, which is very weird. Gnetophytes are dioecious, so again, they're going to have male and female plants. They're both wind and insect pollinated, and some even produce nectar, which is something you would find in flowers in the Angiosperms. Their sperm lack flagella, and they have opposite leaves. And this is, I think, one of the most distinctive characteristics of this group. You don't see opposite leaves too often in many plants. So there are kind of, when you do find opposite leaves, like in the mints or in maples, it's this very distinct characteristic. So I tend to kind of lean toward that one for their identification. Here we have one of our Gnetophyte examples, Welwitschia mirabilis. So this plant can live for thousands of years, and it only has 2 leaves, and they just grow continuously from a basal meristem. So much like we saw in the hornwort sporophyte, instead of having this tip growth, this apical growth, where the youngest parts would be at the ends, the youngest parts are here at the base of the leaf, and they just keep pushing older cells outward. And those older cells can die and dry out, and it keeps growing that same leaf from that basal meristem. And the leaves split as they get kind of larger, and you get this kind of huge stack in the middle. I think if a person was standing here, it would be, gosh, I don't know. I've seen ones where the person is like this big, but this might be a smaller version. These plants can get gigantic, and they only grow in Namibia. If you ever get to go to the HSU greenhouse, which we normally do as a field trip, they have a few great examples of Welwitschia. So these are also dioecious because we're in the Gnetophytes, and they have male strobili and female strobili, and those grow on different plants. This is what the strobili look like. Here is another example of a Gnetophyte. This is Needham. There are many different species of Needham, and most of them are tropical. But they make these angiosperm-like trees, so they look like flowering plants. And you can see that here. These are the male reproductive structures and the female reproductive structures. So this looks very much like a fruit, and this looks very much like, almost like a mint flower, this kind of stack of little floret-looking structures. So this would be microstrobilus. Ephedra is another example of one of our Gnetophytes, and this is one that is at the HSU greenhouse. You can see it has these little scale-like leaves, and these would be arranged opposite from each other. So we would see one here and one here. That would happen at each node. We'd have a little pair of scale-like leaves across from each other. These are the male cones, and these are the female cones. So again, we have these very weird angiosperm-like structures. These look a lot like little florets that you would see on a rush or something. And then over here, we have almost berry-like structures as for our Megaspore, so our Megastrobilus. It's very interesting how much convergent evolution we have with the Angiosperms. Weirdly, when I was reading about the Gnetophytes, ephedra, they release their pollen at the full moon, potentially because they use moths and other nocturnal insects as their pollinators. Very fun. And the Gnetophytes lead us to the Panopto. Taxonomy is always changing as we learn new things with genetics about how groups are related or how we think they're related. So even class to class as I teach this, it used to be Coniferophyta, then it turned into the Pinophyta, and now it's the Panopsida. So just thinking about how we sort of change our ideas about how things are related, and that influences how we have to name them based on naming conventions. So these are typically evergreen trees, but there are examples of deciduous trees like larch or dawn redwoods. Those are going to lose their leaves seasonally. But because we have these xerophytic leaves that we invest highly in, we want to keep them around. So they tend to be evergreen. Unlike the rest of the Gymnosperms, this group is monoecious. So all of our reproductive parts are going to be housed on the same plant. So here we would have our megastrobili, our seed cones, and our microstrobili, our pollen cones, on the same plant. All of our conifers are wind pollinated, so here you can see this huge cloud of pollen being released from these pollen cones. They really mass produce that pollen, and it gives a lot of people allergies. I love the timing of this class because whenever it is time to talk about pollen, there's pine pollen everywhere. So you can just find it coating your cars right now, and it's giving me the sneezes. They're wind pollinated and their sperm are unflagellated. So this is a group where we see this loss of flagella. Here you can see a pine pollen grain, and it has these two air sacs. So this would allow it to sit on top of a body of water or to float better through the air because it has these almost like wings. We're going to draw the pine life cycle, and as you might have noticed, our life cycles are getting increasingly complex. So this is, we'll do our best here. Here's meiosis and fertilization. We know that everything before fertilization is going to be haploid, everything after fertilization will be diploid, so we'll keep them separated on either side of this line. But we'll see that this haploid and diploid tissue is going to be contained within the same structure because we have the retained megaspore. So let's start with a pine tree. That's our sporophyte. So this is our diploid sporophyte here. And that sporophyte is going to produce megastrobili and microstrobili because it is monoecious. So here's my little microstrobilus. It's our pollen cone. And within this structure, there would be a bunch of little microsporangia. I'm going to switch over to the female cone. So the female cone has this ovuliferous scale. That's where the ovules will be produced. And below that is a sterile bract. And this whole structure here is a megastrobilus, or seed cone. So the megastrobilus, you have the megasporangium, and that is surrounded by this tissue called the integument. And I will label this all after we go through meiosis. So inside the megasporangium is the megaspore mother cell. Inside the microsporangium, there would be many microspore mother cells. So microspores, megaspore inside the megasporangium. These are the two sites of meiosis. So microspores are going to undergo meiosis, and the megaspore is going to undergo meiosis. So inside that pollen cone, I'm just going to zoom in on one microsporangium. So there's that microsporangium, still diploid, but within it, we now have the production of these pollen grains. And they don't instantly turn into pollen grains. It's not like you take a microspore and then it goes through meiosis and it becomes 4 pollen grains. It is going to have to grow into that pollen grain because it's 4 cells. So we're simplifying here a little bit. So if we zoom in on one of those pollen grains, this is the microgametophyte, or the male gametophyte. And it has air sacs, which would be much larger than I've drawn them here. It has 2 cells that don't really have any specific function. Those are the prothallal cells. Oh shoot, I shouldn't draw that right there. Prothallal. So you can think of these as like vestigial cells from when we used to have a larger gametophyte, a prothallus. So we have prothallal cells, we have I'll just draw it right here, the generative cell, this is going to make the sperm. And then we have another cell in here, but it takes up basically the entire rest of the space. So we tend to see a nucleus with a nucleolus that looks like a cell, but it's the nucleus of the tube cell. And this grows the pollen tube. So I paused that recording and I gave myself another slide to make this a little bit bigger and less confusing or less cramped. And one thing I did say incorrectly, so I talked about the megaspore mother cell, and that was going to go through meiosis and divide into megaspores. What I didn't talk about, or what I mislabeled, was the microspores. They're not microspores before meiosis, they're microspore mother cells. They're microspores after meiosis, and those microspores grow into the pollen grains. So they divide twice to then become that microgametophyte. So we get microspores and megaspores are the result of meiosis. Okay, so we drew our microgametophyte. Now we need to draw our megagametophyte. So we zoomed in on the last one, and we'll zoom in on this one too. So we have the megasporangium. And we had 4 megaspores get produced within that structure. is surrounded by the integument. And then this hole, this gap in the integument here, is called the micropyle. And then this green is the megasporangium. So of these megaspores, 3 of them die. And only one is going to grow into the gametophyte. So this all happens in those megastrobili, and that's happening on the sporophyte, right? Because the megaspores are retained. So we have our integument with a huge micropyle now. Sorry. The megasporangium is still here, and then our megagametophyte grows by mitosis within that megasporangium and develops 2 archegonia. Usually, sometimes there's more than 2, sometimes there's only 1. So we have 2 archegonia, each with 1 egg. And all of that would have been produced by mitosis. And it's happening still on that sporophyte. So this is all attached to our sporophyte. Our pollen grains get released. And we have wind dispersed, aerial dispersal. So that pollen grain is going to need to land on the megastrobilus, on that female cone. And there's something called the pollen drop that I think it's exuded through the micropyle, and it's this liquid that's going to come out of the cone, grab onto, just because water, you know, it's grabby, it's going to grab onto those pollen grains, and then once it has the pollen, it retracts back into the cone. So the pollen drop has this really important role in making sure that pollen can get to the eggs. So let's draw not the pollen drop, but this whole sort of situation happening here. So we have our integument, and this would all still be attached to that ovuliferous scale. We have our diploid megasporangium, we have our haploid megagametophyte, and everything in that is haploid, the archegonia and the eggs. The pollen grain is going to grow with the tube cell a pollen tube. And these can get crazy long. If you think about corn silk, corn pollen has to grow a pollen tube all the way down one of those corn silk threads to get to the corn egg, which is pretty crazy. So the tube cell grows a tube. That's its whole job. And the generative cell divides by mitosis to make 2 sperm. Those are going to fertilize, or one of them will fertilize one of these archegonia. So we get 2 sperm. And these are deposited in the archegonia by the tube cell nucleus. Or not tube cell nucleus, by the tube cell's tube. That's what I get for talking and writing. So the generative cell is going to divide by mitosis to produce those 2 sperm, and the tube cell is going to grow the tube so that they can get to the archegonia. And we are going to get fertilization, which I'm going to show you what this fertilized seed looks like on another slide, because this here is the ovule, and once it gets fertilized, it becomes the seed. When fertilization happens, when the sperm cells make it to one of those eggs, we get a bunch of changes. So this is the formation of the zygote, but it happens, again, on the sporophyte still. So we have our integument, which now is going to fully close off. So the micropyle closes because it doesn't want to let any more pollen grains in there. Now we're trying to protect what's inside because the job has been done. So the micropyle closes and the integument becomes the seed coat. So once that micropyle closes and it is a complete coating, we have a seed coat. And this tissue is going to be diploid because it is part of that sporophyte. We still have this layer of megasporangium. So that megasporangium never went away. It's still enclosing what used to be the megagametophyte, and the megagametophyte is still there, I should say. So the megasporangium is now going to be called the nucellus. And this is also diploid. Within that, we have the megagametophyte. Which is haploid. And then we have now more diploid tissue because we had fertilization. So that zygote that forms within that is going to grow. This is now, it starts as a zygote, and that's your first diploid cell. It grows by mitosis within this structure, and because it's retained, it's called an embryo. So that embryo grows within the megagametophyte, which is within the megasporangium, which is within the integument, which is held on that seed cone. So this whole protective structure is all to make sure that once fertilization does happen, that we are ensuring that we didn't waste that opportunity, right? So now we have this nice package here that can be released. And it's now, once it's fertilized, it's called a seed. So the seed is released. It has this wing, and then the seed. And that seed contains all of these layers that we just covered. And that embryo is also diploid. So this is kind of continuous with the integument, but it forms this sort of wing. And then that can grow into, oh, thank goodness, that sporophyte that we started out with. And that's a pine life cycle. So as you can see, things start to get really crazy, and the angiosperm life cycle is even crazier. So some things to think about for the pine life cycle and things to note. The ovule consists of the megasporangium and a few layers of surrounding tissue called the integuments. After fertilization, the integuments become the seed coat. In seed plants, the megagametophyte has 2 archegonia, and it grows inside the megasporangium. The megasporangium becomes a nucellus. And this says in seed plants. I don't know why I didn't just say in gymnosperms, because we are going to lose archegonia when we move into the angiosperms. So this really is gymnosperms. And particularly in the pine life cycle. So in seed plants, the megagametophyte grows inside the megasporangium. That's true. This 2 archegonia part is not necessarily uniting the groups. The megasporangium becomes the nucellus, so that is diploid tissue. The embryo develops inside the megagametophyte, which is inside that sporangium. And that embryo gets to consume all the stored food that is the nucellus and the megagametophyte. So those all provide nutritive tissue to feed that growing embryo. So megagametophyte and the nucellus are just sources of food for the developing embryo. And all of the parts that are in bold are components of a seed, and a seed is a fertilized ovule.